Welcome to the Lymphoma Research Foundation's update on the American Society of Hematology meeting webinar. I'm Mark, and I'll be the moderator for today's call. During today's call, you will hear from two expert speakers, and you will have an opportunity to ask questions. If you have any questions during today's presentation, you can ask them at any time in the Q&A box on the webinar. As a reminder, this webinar is being recorded. At the end of the program, a link will appear on your screen. Please follow this link to complete an evaluation of this program and gain certification of attendance. If you are listening by phone, this link will be sent to your email at the end of the webinar. Now I'm pleased to introduce Jesse Brown. Jesse Brown is the Associate Director of Patient Education at the Lymphoma Research Foundation. Welcome, Jesse.
Thank you, and thank you to each of you for taking the time to join us in today's update on the American Society of Hematology meeting webinar. I'd like to thank our sponsors of this webinar, Genentech, Celgene, Foundation Medicine, and Kite, a Gilead company. We have callers from 41 states across the U.S., as well as patients and caregivers from 15 countries around the world. Access to expert disease information is so important, and we are thrilled to be able to bring you this educational program. We have a wonderful program planned for you today, and I'm honored to introduce you first to Dr. Andrew Zelenitz. Dr. Zelenitz is a medical oncologist with special expertise in lymphoma and a former chief of Memorial Sloan Kettering's Lymphoma Service. He is also currently serving as the chair of LRF's Scientific Advisory Board. Dr. Zelenitz, I will now turn the talk over to you.
Well, I want to add my thanks to everyone for joining us today, and thank the LRF for the invitation to do this update about what's new in B-cell lymphoma from ASH, and maybe a little bit more than just ASH. 2019 saw tremendous advances. It's impossible in 15 minutes to summarize these. What I'm going to do is hit some of what I think are important highlights. I'm happy to address other issues in the questions. So let's talk about small lymphocytic lymphoma and chronic lymphocytic leukemia first. And, you know, one of the studies that was presented this year at the International Workshop on CLL was CLL-12. And CLL-12 asked the question that we've been asking repeatedly over the years: can we replace observation for patients with high-risk CLL? So what is high-risk CLL that doesn't need treatment? Well, the risk model is shown on the slide. It includes deletion 17p, serum thymidine kinase levels, serum beta-2 microglobulin levels, whether the IGHV is mutated or not, how well patients are feeling, presence or absence of deletion 11q, male sex, and age greater than 60. Patients who have 3 or more of these factors are considered high risk, and we know that these patients tend to progress to needing treatment earlier than patients who have 0 to 2 of these factors. So we've asked the question a number of times, can we intervene? And one of the interventions was to use a novel agent, that is ibrutinib. What ibrutinib clearly, unequivocally did, and this is not surprising, showed that the time to symptomatic progression was clearly prolonged with initial treatment with ibrutinib. However, there were some interesting factors here. 34% of patients actually came off study on the ibrutinib arm mostly because of side effects. Not too many progressions, but there were many patients, a third of patients, came off for side effects. On the placebo arm, the patients who got active surveillance, about half of them came off and they progressed, and that's not surprising. But importantly, to date, this study, which has been going on for more than 5 years, has shown no difference in overall survival, and I believe that active surveillance remains the standard of care for patients with even high-risk observable CLL. Now, that's different than patients who need treatment. So who needs treatment for CLL? It's patients who have impact on their bone marrow with either a low hemoglobin or a low platelet count, have very large spleens, very large lymph nodes, whose disease is clearly progressing, as evidenced by a rising white count, or have other disease manifestations, including autoimmune complications such as hemolytic anemia or thrombocytopenia because of platelet destruction, or disease-related symptoms such as fevers, night sweats, weight loss. If any of these are present, that's a reason for initiation of systemic treatment, and this is what I think is the paradigm for treatment today in CLL. It includes, for younger patients who have no IGHV mutation and no deletion of 17p or mutation of TP53, the standard of care is FCR, in my opinion, though other treatments are acceptable. Always, clinical trial is an appropriate option in all of these circumstances. For those patients who have a deletion of 17p or a mutation of TP53, then chemoimmunotherapy is no longer appropriate, and here it's either a BTK inhibitor such as acalabrutinib or ibrutinib, or venetoclax and obinutuzumab, again, as a clinical trial. And I'm not going to repeat this every single time, but clinical trials are the heart and soul of making advances in this field. For the patient who's over 65, fit, but has mutated IGHV, again, I think that we have yet to see that bendamustine and rituximab clearly is been bested by BTK inhibitors, even though these are appropriate choices. The reason why I still feel that the chemoimmunotherapy is the best choice is it gives us another option for treatment, which we may not have in the future if we skip it early on. And then for the less fit patient where there's mutation of IGHV, but the patient's less fit, here a less aggressive chemotherapy would be appropriate, such as, and this is a typo, this is supposed to say obinutuzumab plus chlamydyl, or venetoclax plus obinutuzumab are both appropriate, as are BTK inhibitors. So some of this is based on a study that was presented by Jeff Sharman at the ASH conference, where it was called the Elevate Treatment Naive Study, comparing acalabrutinib to a standard treatment for frailer patients of obinutuzumab plus chlamydyl, to a third arm that was acalabrutinib plus obinutuzumab. And this study showed that the acalabrutinib, the BTK inhibitor arms, were clearly superior to obinutuzumab plus chlamydyl. There was an interesting observation that acalabrutinib and obinutuzumab was a little bit better than acalabrutinib. This was not yet statistically significantly different, but there's clearly a trend here. And what's interesting about this is when we looked at similar trials with ibrutinib, rituximab did not add, the anti-CD20 antibody did not add to ibrutinib. So this, because acalabrutinib acts a little bit differently, it's a little bit more specific than ibrutinib, the addition of the anti-CD20 antibody might provide additional benefit. We also saw important update of the MURANO study, which was for patients with relapsed CLL. This study compared bendamustine rituximab to venetoclax plus rituximab for relapsed patients, and this study again showed a dramatic difference in favor of the venetoclax arm. Venetoclax plus rituximab was clearly better than bendamustine rituximab. What was important about this update is now the median follow-up is 4 years. And so virtually everyone has been followed after the end of their chemotherapy, which occurred at 24 months. And we can see that even off the venetoclax, there is a sustained benefit. Yes, there is a decline in the curve, but that decline in the curve is associated with people who did not achieve a undetectable minimal residual disease state. So it turns out that the ability to detect tiny amounts of disease in the blood is a very strong predictor of outcome. And for patients who fail to achieve an undetectable minimal residual disease state, I would recommend that treatment be continued and not stopped at the end of 2 years. Another exciting presentation was from Dr. Siddiqui from the City of Hope, which looked at the role of CAR T cells in the treatment of CLL. CAR T cells are cells where we put an artificial receptor into patients' T cells and promotes enhancement of killing of the tumor. If we look at the response with lisacaptogene maraleucel, or lisa-cel as it's sometimes called, the overall response rate was 82%, with the complete response rate of 45%. In patients who had failed either a BTK inhibitor and venetoclax, there was an equally high response rate, suggesting that this provided an alternative means of treatment, and this is really quite an exciting result. Minimal residual disease was achieved with this approach, so that the vast majority of patients were able to achieve an undetectable state of minimal residual disease. The CAR T cells are associated with risks, including cytokine release syndrome and neurotoxicity. But in this study, which is small and it's just a pilot study, the rate of high-grade CRS was very low, only 9%, and the risk of high-grade neurotoxicity was only 4%. How about mantle cell lymphoma? Well, though there's been a lot of activity in mantle cell lymphoma, the real news at ASH was, again, CAR T cells. ZUMA-2 was a study of a novel CAR T cell from Gilead called KTEX19. It's frequently confused with axicaptogene Cilta-Leucel because it looks exactly the same. It has the same construct, but there's an important difference, and that is that T cells are enriched before they're transduced to ensure that we're not transducing any mantle cell lymphoma cells in the product that gets returned to the patient. 68 patients were treated on this study, including a quarter of the patients having a blastoid morphology. One of the somewhat disappointing things is that only 17% of the patients here had mutation of TP53. This is a very common factor in relapsed mantle cell lymphoma, and so this is a little bit underrepresented in this clinical trial. But again, very, very high rates of response, 93% overall response rate with 2/3 of patients achieving a complete response, and the response durations so far have been durable, as you can see here from the progression-free and overall survival. Obviously, we need further follow-up, but it looks like there are patients who achieve a remission and stay in remission for a prolonged period of time. Again, cytokine release syndrome and neurotoxicity are important events, but they were manageable. They were more common than with lisocabtagene maraleucel, but they were very manageable. What about follicular lymphoma? Lenalidomide has been a drug that's been used for more than a decade in management of multiple myeloma, and has been studied in follicular lymphoma for some time. And we know that this is an active agent. It works both on the microenvironment and on the tumor cell directly. Previously, we had seen the results of the Relevance study, which compared rituximab plus lenalidomide to rituximab plus chemotherapy, and there was the rituximab lenalidomide was as effective, equally as effective as giving chemotherapy. So rather than having to take chemotherapy, patients had the option of taking lenalidomide. Though this is not FDA approved in the Frontline, it is FDA approved in relapse. It clearly shows the activity. But another study, the GALLIUM study, asked whether there was a better antibody in follicular lymphoma than rituximab. And this study compared obinutuzumab plus chemotherapy to rituximab plus chemotherapy and showed that the obinutuzumab actually resulted in better progression-free and time to next treatment compared to rituximab, though overall survival at the current follow-up is not yet different. So this raised the question of, can we use lenalidomide and combine the better antibody and get good results? And Dr. Nastapil from MD Anderson presented this study of obinutuzumab plus lenalidomide, and the results are really quite exciting, with complete response rates that are extremely high, in the 94% range, as their best overall response. And if you look at the progression-free survival, the progression-free survival thus far has been really outstanding, and this is a very exciting result. I will remind you, this is what it looked like in Relevance. So this is a better looking curve, and we're hoping that we're going to see further follow-ups on this exciting data. Well, what about new approaches to immunotherapy? coming back again to lenalidomide, MUR208 as an anti-CD19 antibody. It's been combined with lenalidomide and used in the treatment of relapsed refractory large cell lymphoma in a study that's shown here, where the MUR208 is given weekly for the first 3 cycles, and then every other week for the next 8 cycles, and then twice, and then lenalidomide is given for the first 12 months. The MUR208 continues afterwards. And the results here are really exciting. These results look as good, in my opinion, as any CAR T result in relapsed refractory large cell lymphoma. So this is an exciting opportunity. And another thing I believe that's challenging CAR T cells in large cell lymphoma is what we call bispecific antibodies. Mosun or Mosunetuzumab is a bispecific antibody that binds a T cell on one side and the tumor cell on the other side, and brings them together to activate the T cell and kill the tumor cell. This was presented by Steve Schuster in the plenary session at ASH. Patients received on a 21-day cycle. They initially received small doses of Mosun to overcome the cytokine release syndrome, and then received drug every 3 weeks thereafter. And in both aggressive lymphoma and in indolent follicular lymphoma, we're seeing interesting activity. The activity is actually more exciting in the indolent lymphomas than the aggressive lymphomas, but even among the aggressive lymphomas, we're seeing a complete response rate of about 20%. These are early days about learning how to use this agent. So these are some of the aspects that, as I said, these are just some highlights. There's lots more that happened, but I'm going to turn it over to Dr. Porcu to share some of the highlights in the T-cell lymphoma.
Thank you so much, Dr. Zelenitz. I'd now like to turn the second part of our webinar discussion over to Pierluigi Porcu. Dr. Porcu is a medical oncologist with special expertise in T-cell lymphomas. He is the Director of Hematologic Malignancies and Stem Cell Transplantation in the Department of Medical Oncology at Thomas Jefferson University. Dr. Porcu, I'll now turn the talk over to you.
Thank you very much, Jesse. Thanks, everyone, for calling in. It's good to be on this program with the LRF, as well as with Dr. Zelenitz. The ASH updates on T-cell lymphoma are going to really focus on a number of facts on the landscape of T-cell lymphomas, which, unlike the landscape in B-cell lymphoma, which has been defined fairly well over the past 15 years, in T-cell lymphoma is really still evolving. So there were a number of abstracts, both oral presentations and posters in T-cell lymphoma that were presented, and I have a list of those in the next couple of slides. But one of the things that also happened at ASH, one of the Friday symposia just before the official beginning of ASH, focused on T-cell lymphoma. The 3 speakers were Dr. Francine Foss, Dr. Barbara Pro, and Dr. Steve Horwitz. And they really provided a very nice overview and survey of the status of the art, essentially the state of the art in T-cell lymphoma. Some of those some of the things that were presented are really relevant for the audience, because in T-cell lymphoma we're still in the phase where the very first steps in terms of making a prompt diagnosis, making an accurate diagnosis, and then the risk stratification, kind of framing the story from the standpoint of the patient coming in, still really remain one of the fundamental tasks that the medical oncologist has to do. And that is in part because of the fact that T-cell lymphomas, as shown in the slide, really represent a relatively small minority of all the different non-Hodgkin's lymphoma. What this slide shows, that if you look at all the cases of lymphomas that were diagnosed in 2019, just about 82,000. Only about 10 to 12% of those were T-cell lymphomas. Now, within that slice, you have to understand that there are at least 30 different types of T-cell lymphomas. So, because of the rarity of these diseases, sort of having really a efficient and accurate diagnosis, it's a complicated affair. And one of the things that came out of the symposium was the fact that you really want to make sure that whenever you have a biopsy with a possible diagnosis of T-cell lymphoma, you really want to make sure that the biopsy actually is reviewed by an expert hematopathologist that has seen a lot of those cases and is able to make the diagnosis. This slide shows kind of the breakdown of all the major different types of T-cell lymphomas. This was done based on the data from a relatively old study, one of the very first studies to kind of look at the landscape of T-cell lymphomas. This was published in 2008. One important thing to remember, because these data are frequently shown, is that the relative frequency of all these different diseases based on the study is probably quite different from the relative frequency of T-cell lymphomas in the U.S. For example, in this slide, you can see that in the green slice, almost 10% of the cases were a type of T-cell leukemia and lymphoma that is quite rare in the United States. And another very large slice is based on extranodal and K lymphoma. It's the red slice right there at 6 o'clock. Now, fortunately, we're starting to see now data collected, population-based data collection in T-cell lymphoma on registries in the U.S. And this is the study that has the largest amount of data so far. It's called the Complete Registry. And as the slide shows here, you can see that peripheral T-cell lymphoma, not otherwise specified, really represents the majority of the case of T-cell lymphomas. And there are, angioimmunoblastic T-cell lymphoma on the purple slice there represents another significant fraction, and then the anaplastic large cell lymphoma. So between the four of them, these four types of so-called nodal T-cell lymphoma, they represent the vast majority of the cases that any oncologist will see. And then there's a whole spectrum of rare types, which again require a lot of expertise to be diagnosed and then treated. This slide shows the current classification of the World Health Organization on so-called T-cell neoplasms, because it includes both leukemias and lymphomas. And of course, on one side you have the precursor type, which are mostly ALL, acute lymphoblastic leukemia and lymphoblastic lymphoma. And then there's a very large number of different so-called mature T-cell neoplasms that are listed according to the clinical presentation. This remains a very important component of making a diagnosis in T-cell lymphomas. These are certain category of diseases that are predominantly involved in the skin. Another set of diseases that represent mostly focus on extranodal sites, or outside of the lymph nodes. And then you have the predominantly lymph nodal-based T-cell lymphomas, which also can have fairly extensive extranodal involvement. And then there is a category of T-cell neoplasm predominantly affecting the blood. Some of these diseases are associated with viruses. That's something else that is really becoming sort of clear. And one of the viruses in yellow is called HTLV1, which was the very first oncogenic virus really kind of proven and in T cells. And another virus is the Epstein-Barr virus, which tends to affect predominantly B cells, but also involves T-cell lymphomas, as I will show you in the next few slides. So this is just a list of the different presentations at ASH. I'm not going to go through all this slide, but this is provided for the audience so that it can go back as a reference. The first abstract that was impacted and presented is a follow-up on the Echelon 2 clinical trial. As many of you may know, the Echelon-2 was a randomized phase 3 clinical trial that was presented last year at ASH and was published more recently, and essentially has changed the standard of care for patients with peripheral T-cell lymphomas that are expressing CD30. Brentuximab Vedotin is an antibody-drug conjugate that it targets CD30. This particular study that was presented by Dr. Savage at ASH last year essentially is a subset analysis of the Echelon-2 study. And this is based on the idea that perhaps patients treated with brentuximab and CHP may potentially not need to receive stem cell transplantation as a consolidation, something that we routinely do for most patients fit and eligible after chemotherapy with peripheral T-cell lymphoma. The vast majority of the cases of the population in this study were patients with anaplastic large cell lymphoma, and therefore a significant fraction of this study was this subset analysis was focused on anaplastic large cell lymphoma. But nonetheless, both for the ALK-negative anaplastic large cell lymphomas and for the non-anaplastic large cell lymphomas, which include the peripheral T-cell lymphoma, NOS, and angioimmunoblastic T-cell lymphoma, there really was a trend towards a better progression-free survival with stem cell transplantation. The patients in the study were allowed to go to stem cell transplantation according to the preference of their treating physician. This was not a randomized study in any way looking at transplantation. Nonetheless, if we look at the numerical the number of patients who progressed with stem cell transplant versus those who did not receive stem cell transplant, it seems to suggest that stem cell transplantation may still be beneficial in this patient population. Certainly, the study did not remove the question whether stem cell transplant really is necessary or not, a question that remains to be proven in randomized clinical trials that are ongoing. This is just a schema of the Echelon-2 trial. The patient needed to have at least 10% expression of CD30 on their peripheral T-cell lymphoma, and they were randomized to brentuximab vedotin with CHP versus CHOP, which is the standard of care. All patients had an end-of-therapy PET scan, and those who were in remission were they were allowed to go to stem cell transplant if their oncologist and the patient thought that was the best way to go. A second study which our group presented was on a new treatment for a subset of T-cell lymphomas that express EBV, the Epstein-Barr virus. So a number of lymphomas, not just T-cell lymphomas, but B-cell, T-cell, and NK, and Hodgkin lymphoma express EBV. Most of the data about the impact of the presence of EBV in the tumor tissue on outcome are retrospective, and therefore they have to be taken with a grain of salt. But there is a significant cumulative set of data to suggest that EBV actually has an impact on prognosis, and therefore potentially targeting the virus is valuable. So this was a study looking at a combination of two drugs. One is an HDAC inhibitor called nanatinostat, and the second drug is a commonly used antiviral drug called valganciclovir. And the goal here was to sort of treat the tumor containing the virus with nanatinostat to activate a certain set of viral products that then make the cells more sensitive to valganciclovir. This was a phase 1/2 that we presented the data for the phase 1 here. So the phase 1 part with 25 patients. This is the schema. Patients were treated continuously with the oral combination. They had intermittent PET scans for assessment of response. The primary goal here was safety primarily, but also trying to get a signal of efficacy. The safety for this combination is really mostly hematological, as expected, and after a number of dose adjustments with the combination, we were able to identify a safe recommended phase 2 dose. The patients included a variety of different types of lymphomas, as shown on the right here, with some B-cell lymphomas, T-cell lymphomas, and Hodgkin lymphomas. Responses really were seen across all the different types. For the audience here today, I think that one of the points of interest is the fact that there were a number of responses seen in T-cell lymphomas with this combination. One of the patients with T-cell lymphoma actually had romidepsin refractory disease and responded to this combination with nanatinostat and valganciclovir. So while this is very preliminary data, I think that there is an interest in actually continuing this study, particularly the safe dose that we identified. Another very interesting study was presented by Dr. Horwitz on the phase 2 clinical trial with this new drug called Cerdulatinib, which is a dual SYK-JAK inhibitor. Now, SYK and JAK are molecules that tend to drive proliferation in T-cell lymphoma. This was in relapsed and refractory patients, both with peripheral T-cell lymphoma and cutaneous T-cell lymphoma. This was a fairly sizable trial with 61 patients with PTCL and 37 patients with CTCL having received at least one prior chemotherapy. The drug is an oral drug given twice a day. And essentially, this really kind of showed, first of all, was a safe drug to use. No major unexpected adverse events were seen, with the exception of some elevation of the pancreatic enzymes without clinical evidence of pancreatitis. But in terms of the efficacy, the overall response rate was 35% in the PTCL as well as in the CTCL. But one particular subtype of T-cell lymphoma, angioimmunoblastic T-cell lymphoma, or peripheral T-cell lymphomas with a follicular helper T-cell phenotype, the response rate was high. This seems to be a trend for a lot of the new drugs in peripheral T-cell lymphomas, where angioimmunoblastic T-cell lymphoma seems to have a higher sensitivity. I think one important thing here is that the responses are also seen in relatively rare and typically chemo-resistant types of T-cell lymphoma, such as gamma delta T-cell lymphomas. So that was an exciting study. I think it's a very promising drug. On the other hand, there were some disappointment in this study presented by the Mayo Clinic group with nivolumab, which is an anti-PD-1 inhibitor, a drug that is approved for Hodgkin's lymphoma and is very active across a spectrum of lymphomas. The Mayo Clinic team conducted a phase 2 study of single-agent nivolumab in patients with relapsed refractory PTCL. The doses of nivolumab was fairly standard, and the primary endpoint here was looking at response rates. Because of the response rate, while the response rate really crossed the threshold that was necessary to move on to the next phase, nonetheless, there were some side effects, and there were also cases of hyperprogression. This is something that has been seen in other type of lymphomas with these PD-1 inhibitors, and therefore there was not a whole lot of enthusiasm in continuing the study. So this it's an issue with the new immunotherapies and checkpoint inhibitors in peripheral T-cell lymphoma. For a variety of reasons, that approach is not as straightforward as it may be in other type of lymphomas. Now, the recommendation from the study was that checkpoint inhibitors should not be studied as a single agent, or they should be cautiously studied as a single agent in T-cell lymphomas, and therefore looking at combination. This is just an example. There are many combinations with checkpoint inhibitors ongoing right now. This is just an example of a trial that they were conducting in collaboration with the University of Colorado in Denver from one of my fellows, who is now there, a combination Frontline patients with Nivo and EPOC chemotherapy, which is ongoing. Finally, another drug that is of interest is tipifarnib, which is it's an old drug. It inhibits a enzyme that is called farnesyltransferase, which activates a series of signaling molecules in the cell. It's not clear that the mechanism of action of this drug, as described, is relevant for the T-cell lymphomas. Nonetheless, in this study from a group led by Dr. Witzig from the Mayo Clinic, also showed a very significant response rate. And again, particularly in patients with angioimmunoblastic T-cell lymphoma. One of the interesting things about this study is that the group seems to have identified certain biomarkers that are associated with a better response to tipifarnib, one of them being this KIR3DL2 genetic variant, and also potentially the expression of CXCL12, which is a chemokine ligand. So this potentially opens the door to biomarker-driven studies in T-cell lymphoma. That is something that we have been missing. Finally, just one word about a study that was not presented at ASH, but was presented at ASCO earlier this year. It's important to mention it because this is a large randomized clinical trial looking at autologous stem cell transplantation versus allogeneic stem cell transplantation as a consolidation. Now, I think that most of us practicing in lymphoma and T-cell lymphoma do not use allogeneic transplantation frontline as a consolidation for patients. We use autologous transplant, but it's good to see that the data from this study, actually randomized study, support that choice. Patients here were randomized to receive either autologous stem cell transplantation or allogeneic transplant after a fairly intensive initial chemotherapy with CHOEP given every 14 days, followed by DHAP. And if you look at both the event-free survival curves as well as the overall survival curves, really there was no significant difference. The reason for death were different, with the allogeneic patients having more toxicity from the transplant and the patients receiving autologous having more progressions due to progressive disease. So the conclusions here my conclusion from ASH this year is that clearly there's a new frontline standard of care with Brentuximab, Vedotin, and CHP for CD30 expressing peripheral T-cell lymphoma. But whether or not autologous stem cell transplantation can be avoided after that is not clear at this point, and the data need to be stronger and confirmed. There are a number of multiple promising new agents in phase 1 and 2 development in clinical trials that are very good. EBV, we believe, is a new target of therapy in peripheral T-cell lymphoma and deserve to be continued to be studied. And then the role of immunotherapy with checkpoint inhibitors needs to be further defined. With this, I'm going to turn the conversation back to Jesse. Thank you so much for listening. Appreciate it.
Thank you so much, Dr. Porcu. Before we open the program to questions, I want to share information with you on the Lymphoma Research Foundation that may be helpful to you as you navigate lymphoma and long-term survivorship. The Lymphoma Research Foundation, LRF, is the nation's largest nonprofit dedicated exclusively to lymphoma. Our mission is to eradicate this disease through investment in the most promising lymphoma research and to serve those impacted by lymphoma through quality education and support opportunities. As we continue to make progress in advancing lymphoma research, we also want to ensure that you have access to latest information about your disease. The Foundation provides comprehensive disease and treatment-specific resources, programs, and services, all of which are offered free of charge and have been reviewed by lymphoma experts. Most relevant to today's call, LRF offers a variety of lymphoma-specific resources, many of which you can access at the bottom of your screen if you are utilizing the web link or via LRF's website at lymphoma.org if you are on your phone. The LRF helpline can answer your specific questions about lymphoma, as well as discuss relevant treatment options and clinical trials. We also offer the Lymphoma Support Network, which is a one-to-one peer support program for people with lymphoma and their caregivers. The Lymphoma Support Network connects patients and caregivers with volunteers who have similar experiences to help give others strength to meet the challenges they may have to face. We also offer a variety of publications that have been reviewed by lymphoma experts to ensure you have access to the latest lymphoma information. We have comprehensive books on understanding non-Hodgkin lymphoma, Hodgkin lymphoma, chronic lymphocytic leukemia, and the transplantation process. We also offer a variety of easy-to-understand fact sheets on each of the subtypes of lymphoma, as well as current treatment modalities, including a dedicated CAR T-cell fact sheet, as well as supportive care topics that may be relevant to your lymphoma experience. The Lymphoma Research Foundation also hosts in-person education programs across the country to provide an opportunity for you to learn from and ask questions of lymphoma experts who are at the forefront of some of the research that you heard about today. I would encourage you to visit our website, lymphoma.org, to find out if there is a free educational program in your area. Finally, our mobile app, Focus on Lymphoma, is an award-winning app that provides patients and caregivers access to comprehensive content, as well as unique tools to help manage your disease. The app is available for free download in the Apple App Store and in Google Play, and I would encourage you all to download the app today. And last but certainly not least, for those of you who may be looking to give back and help continue to advance lymphoma research, I would encourage you to join Team LRF. We offer a variety of walks and rides for you to participate in, or you could turn your talents and passions into ways to raise funds for lymphoma research. Through Team LRF, you can meet others who have been impacted by this disease and join a community of individuals committed to impacting lives by finding cures for lymphoma. I really hope that you will take advantage of some of the great resources and services that the Lymphoma Research Foundation provides. If you have questions regarding what you learned today, or you need information about relevant treatment options and supportive care resources, you can reach out to the Lymphoma Research Foundation through our website or by calling our helpline at 1-800-500-9976. Thank you all again for your time today. We will now begin our Q&A portion of the program. Just as a reminder, please keep your questions as general as possible so that the entire audience can benefit from Dr. Zelenets and Dr. Porcu's answers. And with that, I will turn the call over to our moderator.
If you would like to ask a question, please press Submit in the Q&A box on your screen. We will take as many questions as possible, but if you have a question that does not get answered, you can always reach out to the Lymphoma Research Foundation's helpline at 800-500-9976. We have our first question. What are the treatment options for patients who have failed CAR T cell therapy?
So I can address that. This is Dr. Zelenets. So the the MOSIN trial that I presented actually included 30 patients who had had previous CAR T cells. Now, if you remember, mosunetuzumab, or MOSIN, which is why we say MOSIN because it's impossible to say the other, is a bispecific antibody that binds CD20 and CD3. So we're bringing T cells in context to B cells, but most CAR T cells available today in the market are, again, for large cell lymphoma, are against CD19. So this is an alternative target, and there was a 20% response rate, but importantly, a 15% complete response rate, many of which were durable. So there's a lot of work to be done in the CAR T refractory population, but unfortunately these represent very aggressive and very difficult tumors. But I think we're starting to address that with some of the newer agents.
And our next question, how can patients join registries if they are interested?
I can address that. Registry studies are just like any other clinical trials that are managed through the clinical trial offices of each cancer center. Often registries are also available in large community oncology practices. So my suggestion is you can do several different things. One is, depending on the type of lymphoma you have, do a search, or actually talk to the folks at the LRF, and they can help you out identifying the registry sources for the type of lymphoma that you have. And then once you have that information, try to find the closest place to you where that particular registry is open. If you don't do that, ask your medical oncologist. There are a number of different opportunities, and these studies are now being sort of kind of deployed really across a lot of the population in the United States and abroad.
This question is directed to Dr. Zelenets. I'm still on watch and wait for my lymphoma. When would it be necessary for me to look into clinical trials?
Excuse me, can you repeat the first part of the question?
Sure. I'm still on watch and wait for my lymphoma. When will it be necessary for me to look into clinical trials?
So there are a number of diseases that for which active surveillance is appropriate. marginal zone lymphomas, follicular lymphoma, chronic lymphocytic leukemia, small lymphocytic lymphoma, some forms of mantle cell lymphoma. Generally, observation continues until there's a reason for symptoms. However, as I showed in the CLL-12 study, there are some people asking questions about whether outcomes can be improved for patients who have low disease burden, low tumor burden disease, and have studies such as the CLL-12, which was active treatment versus continued active surveillance. So depending on the type of lymphoma you have, there may be a clinical trial appropriate even in the active surveillance phase of the disease. However, more typically, clinical trials are going to be available once treatment is needed, that is, if there are symptoms or there's other evidence of progression, bone marrow compromise, and if we see evidence of progression, then clinical trials will often be available at all phases of the disease, when first treatment is needed, and as well as at relapse. The questions that we ask at different points in the disease are different. In that first-line therapy, we ask questions like we did in GALLIUM or in Relevance. Can we improve on a standard therapy? Can we make it better? Can we replace more toxic drugs with better tolerated drugs? But clinical trials, are widely available in all different parts of the disease process, whether it's from initial treatment to second-line treatment, third-line treatment, and beyond.
Our next question is directed to both speakers. At ASH, were there any research updates regarding classical HL, and what options are available if immunotherapy initially works but stops being effective?
I'll take a crack at this, and then I'd like to see what Dr. Zelenis has to say about this. But it's so immunotherapy, of course, is very active in classical Hodgkin's lymphoma. But as the questioner implies, and is true, that patients tend to progress even after that. So the number of patients that are now being sort of seen in different cancer centers and enrolled on clinical trials with classical Hodgkin lymphoma who have received immunotherapy is becoming a very significant fraction of the patients. So fortunately, there are a lot of new clinical trials that are addressing the patients who have relapsed Hodgkin lymphoma after immunotherapy, including some CAR T cell trials that target CD30. So we're talking about small molecules drugs. We're talking about other combination immunotherapy. It's a need for sure. It's a group of patients that still remains that clearly needs additional therapy. But there's a lot of work now being done addressing that particular need. I don't know if Andy, you have anything to add, or you want to say something about that?
So I agree. Like with many other drugs, we're trying to figure out how to better use the immune checkpoint inhibitors, and we're seeing them incorporated into earlier phases of treatment. We're seeing them combined with other drugs so that we increase the number of cured patients so that there are fewer patients with relapsed refractory disease. But for patients who are currently receiving immune checkpoint inhibitors and then experience disease progression, there was a preliminary, actually I think it was a poster about the trial itself, but there was some information about adding an HDAC inhibitor, tinostat, with checkpoint inhibitors. HDAC inhibitors upregulate the targets for the checkpoint inhibitors and potentially can resensitize patients. And there's an ongoing trial of atinostat and checkpoint inhibitors. Also from Carlos Ramos, updated his experience with an anti-CD30 CAR T cell, showing some favorable responses with CAR T cells, and a number of the patients in that trial were refractory to checkpoint inhibitors. So, so as we develop new agents, they're going to be tested in exactly the patients for whom the current therapies are no longer as effective as they were.
Our next question is for Dr. Porcu. Do you envision CAR T-cell therapy for T-cell lymphomas in the future?
So the answer is yes, but it's going to take a while, and I think we're still at the drawing board phase here for T-cells. The primary reason is that the same cells that are being used to actually develop the treatment product, they have the same markers, the same phenotype of the tumor cells. So there's a lot of what is called, in lay terms, it's called fratricide, so that some of the CAR T cells are actually potentially attacking some of the T cell, the normal T cell themselves. I think that there are a number of astute and creative ways that biotech companies and cancer centers are looking at developing other type of CAR T cells. So I'm sure is going to come to fruition, but right now there's nothing really further along, and I'm not sure that I see any winning horse yet in this race. But there's a lot of very good people are working on it, and so I think we're going to see something good, with the exception of the CAR T against CD30 that Dr. Zelen has just mentioned, that can be applied to a variety of T-cell lymphomas as well.
Our next question is for both speakers. Are there any updates reported for the treatment of double-hit lymphoma?
Andy, you take that.
I can take that. Double-hit lymphomas remain a challenge. Some of the good news is that some of the more recent retrospective data suggests that the outcome of double-hit lymphomas is not as grim as was first reported. And honestly, I can't explain why that would be, but certainly the long-term outcome of patients with double-hit lymphomas is looking more favorable. Not quite the same as otherwise de novo diffuse large B-cell lymphoma, but not as grim as we saw. The standard of care remains dose-adjusted EPOCH. However, I would say that we really need to see some real advances. Last year at ASH, Frank Morhauser and myself presented data from the Cavalli study where we combined venetoclax, an anti-BCL2 drug, with CHOP chemotherapy. and that actually looked very favorable in the double hit population. So the CLGB is studying this in combination venetoclax with dose-adjusted EPOCH, and this is an ongoing study, so obviously it wasn't reported at ASH, but is an important question given the results of the Cavalli study. So we're hoping that we're going to make progress, but we certainly, this has been a difficult area because it's been very difficult to target MYC, which is one of the two important proteins, one of the key proteins overexpressed in double-hit lymphomas.
Our final question is for both speakers. For a relapsed mantle cell lymphoma patient that's 6 years post-R-CHOP and autologous stem cell transplant, what is your preferred approach for the next treatment?
My approach would be would be a clinical trial. I actually, one of the most interesting clinical trials that recently was completed, and we'll see at the next ASH, not at this one, is a combination of ibrutinib and venetoclax compared to ibrutinib alone to ask, is that combination superior in the relapse setting for mantle cell lymphoma? Outside the setting of a clinical trial, it depends on whether or not there's evidence of a TP53 mutation. If there is, then I think a ibrutinib or venetoclax-based approach is the most appropriate. If there's no TP53 mutation, I would use rituximab lenalidomide or obinutuzumab lenalidomide in that setting. So there are many options for treatment in a setting like that because that's a relatively favorable setting to have relapsed late after the high-dose therapy and stem cell rescue.
Thank you again to each of you for joining us on today's call, and we hope you found the information both informative and also hopeful. Please remember, if you have any additional questions or would like to be connected with someone else who has been impacted by lymphoma, you can reach out to the LRF helpline at 800-500-9976. Also, at the conclusion of this program, you will receive an email prompting you to complete a program evaluation. I would ask that you please take a few moments to complete this brief evaluation, as they are really important for helping LRF to ensure they deliver the most useful and meaningful programming to you. And with that, I would like to thank you all again for joining us, and have a wonderful day.